Hello, everyone. So this is the fourth video uh, for chapter 11, and we will cover here section 11.4 on a phenomenon called non-disjunction, which is intimately related to errors in meiosis. Your reading assignment for this section is found in pages 238 to 240, 241 actually, because that page includes the self-assessment question page. Okay, so the main learning objective for 11.4 is for the student to predict the possible changes in chromosome number due to non-disjunction. So the main effect of non-disjunction are alterations in chromosome number. So your objective, therefore, is to learn how this happens. So the sub-objectives would be to describe the effect of the failure of chromosomes to pair or separate properly during meiosis. Second, we need to predict the effects of non-disjunction during meiosis on chromosome number in gametes or in sex cells. Third, you need to describe the detrimental effects of extra or missing chromosomes. Fourth, you need to define the chromosomal makeup of individuals with Down syndrome. And fifth, you need to describe the effects of missing or extra sex chromosomes. So to assess your understanding of this section, please answer the two following questions. How does non-disjunction in meiosis 1 or 2 result in extra or missing chromosomes? Try to make a diagram to answer the question. 12, a male baby is born with the sex chromosomes constitution XYY. Both parents have normal sex chromosome, XY in father, XX in mother. In which meiotic division of which parent did the non-disjunction take place that produced the XYY baby? So what is non-disjunction? Non-disjunction is a form of error in the process of meiosis, which can lead to cells with either extra or missing chromosomes. And specifically, non-disjunction is the failure of a pair of chromosomes. Remember those bivalents that are separating during metaphase 1, or even failure during metaphase 2. The failure of the pair of chromosomes to separate during anaphase of cell division would result in gametes that either have an extra copy of the chromosome or no copy of the chromosome. Non-disjunction that takes place in the first division of meiosis is more common than non-disjunction in the second division of meiosis. So let's have a closer look at this first and second division and this non-disjunction that I just mentioned. Now, in the first division as non-disjunction, what happens here is that the bivalents, which is shown here in meiosis 1, and remember bivalents are paired homologous chromosome, failed to separate from each other during the process of anaphase, so that the bivalent chromosomes actually co-segregates together on one side, on one end, and then leaving the one cell, the other end, without the homologous pair. Of course, this happens within the context of many different bivalents that might be separating together normally. So this is usually a case where only a pair becomes defective in terms of the separation of the homologous chromosomes. So if this happens, the pair of chromosome that went through non-disjunction will therefore create daughter cells at the end, which that will have an extra chromosome after meiosis 2, or gametes that are missing a chromosome. Now, if the process of non-disjunction happens later, and in other words, happens in anaphase 2, so let's just assume that anaphase 1 happens normally. So as you see here, this is a bivalent chromosome pair that has separated normally in anaphase 2. However, at anaphase 1, however, at anaphase 2, okay, it is quite possible that the sister chromatids, it no longer the bivalents, but the sister chromatids fail here to split up. So something happened in the centromere that probably that did not divide, or maybe they have split up, but the spindle microtubules have a defect and allowed both sister chromatids that have become now independent chromosomes to co-segregate together on one side and left the other side missing a copy. So in this particular case, you end up with 50% normal, because you have the normal set of chromosomes, whereas in the 50% of the cases, you will have a 20% probability that you'll have an extra, and then 25% probability that you have a missing chromosome. In either case, these two are abnormal, okay, and may lead to problems. Obviously, this in 99.9% of cases, whenever you have missing chromosomes, this is definitely deleterious or won't survive. In extra chromosomes, an individual that is derived from these gametes, baby born as a result of fertilization with these gametes, may actually have problems surviving. Okay, so essentially, non-disjunction leads to errors in meiosis that actually cause abnormalities in surviving adults. So if you compare, therefore, the severity of the effect of non-disjunction at the first division versus non-disjunction at the second division, you will notice that first division non-disjunction has more severe effect because all of the daughter cells are abnormal. 100%. Whereas in the second division non-disjunction, 50% of the gametes would be normal. One of the familiar disorders that we know of that is a result of errors in meiosis due to non-disjunction is called trisomy 21, or so-called Down syndrome. In the United States, approximately about one every 700 births manifest this kind of disorder. And here what you see is a karyotype. We have seen already what a karyotype is in section, I believe it's 11.2. And this is just a display of all of the chromosomes in the cells of an individual. And what you see here is, if you look at all of the chromosomes in the diploid set for people that are affected by trisomy 21, is that you have an extra chromosome 21 in their cells. And these individuals appear to have a very specific sets of clinical traits that is indicative of the condition. And one of the really alarming trend is especially for the mothers, because there is a very clear effect of mother's age on the risk of having a child with Down syndrome. So this has already been well established. So for example, for women that are of 30 years old, there is 1 out of every 900 chance of having a child that has a Down syndrome. If you are 40 years old, there's about 1 in 700 chance. And I believe that if you are in 50 years old, there is 1 in 10 chance. So please take note of this, especially those of you who plan to have families. There is a risk, of course, if you plan to, for mothers who plan to have babies much older in their age. Okay, and what is the reason for this? Why is it not manifested in men, for example? I think one of the reasons is that the way meiosis happens in the development of the egg is actually very different from those of the generation of sperms, because in mothers, actually meiosis starts at a very early age when the female is still very young, and in fact, while it's still a baby, the oocytes, what we call the primary oocytes, enter into prophase 1 and gets arrested, while the female is still very young. And then, of course, during maturity, at every year, these cells, primary oocytes that get arrested, then enters and completes meiosis. So there is a huge delay or gap between prophase 1 and the ensuing stages of meiosis, that the risk of having a microtubule, spindle microtubule that is defective is very high in females. Children born with an abnormal number of chromosomes that does not involve the sex chromosomes, such as trisomy 21, are very rare. But extra or missing sex chromosomes, on the other hand, either those that involve the X or the Y, are relatively common. It appears as if the X or Y chromosome changes or abnormalities have fewer effects than the extra chromosomes that are not sex chromosomes. The non-sex chromosomes, by the way, are called autosomes. Just for you to know the technical term. So like example of this is the chromosome number 21. Okay, so all the 22 chromosomes that we have that is not sex chromosomes are called autosomes. Okay, whereas the sex chromosomes are simply called sex chromosomes. Okay. And in humans, there is the X chromosome and the Y chromosome. And there are, of course, known clinical cases of this. One of the more common examples are the Klinefelter syndrome, or the syndrome that manifests the presence of an extra Y in the individual. And there is the Turner syndrome, where you only have one sex chromosome. So this is an X, or sometimes designated as X0. These karyotypes of XXY are really interesting, because not the karyotypes, the conditions, or the physical conditions of these XXY karyotypes do display physical and or mental effects. And in Klinefelter syndrome, these are all of these lists of manifestations that you see, including physical and mental characteristics that includes tall stature, small testes, and some degree of mental impairment. And the affected males, in fact, do not produce sperm, and therefore are sterile. Okay. Now, in Turner syndrome, the affected individuals have the karyotype X, and physical and mental characteristics could include short stature. They have some characteristic facial features. They have what we call a neck skin webbing, constriction of the aorta, poor breast development, widely spread nipples, underdeveloped gonadal structures, and in fact, they do not menstruate. Okay, so you can therefore see that any kind of non-disjunction process that happens in meiosis can lead to disorders with varying degrees or spectrum of clinical manifestations or abnormalities. Okay, so this is the end of this section. I hope that you appreciate how important it is for the spindle microtubule to be able to guide and regulate the separation of homologous chromosomes or even individual chromosomes to separate them into daughter cells in order to maintain the proper chromosome number, and therefore the complete set of genes that are required for healthy, full biological development. Okay, so to end this section, I would like you guys to consider this REEF question. This is actually a very challenging one, and I hope that you spend time to figure this one out by writing things down on paper. Okay. So the most common form of hemophilia is a defect in blood clotting factor 8, which is caused by a mutant form of a gene on the X chromosome. Boys who inherit that mutation from their mothers suffer from uncontrolled bleeding. Wow. Girls carrying one copy of this mutation have near normal blood clotting. And commonly, a girl is born with hemophilia even though both parents have normal phenotypes. Now, which of the statements might explain hemophilia in a girl born to parents with normal blood clotting phenotypes? So you have all of these choices here. And the way to answer this question is figure out whether the non-disjunction happens in meiosis 1 or in meiosis 2. And so make sure that you spend some time. It's worth your effort to figure this one out. Okay, so thank you, and I shall see you in the next and final video for chapter 11.